All right, everyone, thanks for coming to this year's Coven seminar. We're very lucky to have Dr. Plos Biol here. Plos Biol is the Director of Research at the French National Center for Scientific Research, or CNRS, as well as the Director for Ecology and Evolutionary Biology section at the Institut de Biologie de l'Eau. Obviously, I never took French in Paris, where he's also the head of the Planetary Genomics Laboratory. He's originally from the UK, and he completed his PhD at the University of Kent in Belgium, followed by postdoctoral research at the Rockefeller University in New York. Then he went back to Europe, where he was started studying plants and diatoms at the Station Zoologica in Naples, Italy, before moving to his current position in Paris in 2002. Again, he's done a lot of work with plants and uncovered roles of superoxide dismutases, response to environmental stress, and key players in light signal transduction. But beyond plants, he's done a great deal of work on diatoms, including establishing molecular tools to study gene function and coordinating several whole genome sequencing of several species. This work has led to a lot of revelations about diatom responses to nitrogen, iron, and light. He's also the coordinator for the evolutionary genomics section of the Tara Oceans project, which is what he's going to talk to you today. And we're very lucky that he's currently on sabbatical at Harvard as a Radcliffe Institute, or a Grass Fellow at the Radcliffe Institute. And without further ado, he's going to talk to us again about Tara Oceans, ecosystems, biology at planetary scale.
Thanks very much for the introduction, and thanks for the invitation to come here. It's a great pleasure to be here, and I'm honored to have been chosen by the students or something to not sure what you were getting yourselves into. Yeah, it's great to be here, so thank you for that. So I will talk to you today about the Tara Oceans project, give you a bit of introduction if you're not really familiar with it, and then go through different results that we have that we have obtained over the last few years. So the Tara Oceans project is to some extent unusual because the nucleus of the project is a sailboat. It's a sailboat equipped for research. You see here, called Tara, 110 feet long, equipped for like 14 people on board. So there'd be like half scientists, half sailors who are on board at any one time. And this is the project that this is the boat that we used to run the expedition. That was the base of the project that I'm going to talk to you about. So the focus of the project. Well, I should first tell you that the Tara research vessel is owned by a foundation. The Tara Foundation for Marine Research, which is owned by a fashion company called Agnès B, based in France. So she graciously gives money for scientific research by lending this boat out to scientists who she thinks have important sort of environmental work to do. So she was really foundational in getting this project off the ground, as was her son, Etienne Bourgois, who runs the Agnès B company, and Eric Karsenti, who is our scientific director, who's a cell biologist, not a marine biologist, he's a cell biologist from the EMBL in Heidelberg in Germany. So he was really got this project going in the first place. The whole subject of study of the project were the humble plankton, which we can consider as the sort of the invisible multitude of organisms. I guess you don't need much introduction to plankton in this audience, but plankton are basically, we can consider as the invisible multitude of life in the ocean. If you take a microscope and you look at the microscopic life and you add it all up, you get to at least a number of like 50% of the biomass in the ocean as being this microscopic life. Okay, it's probably quite a lot more than that, but I think 50% is a very conservative estimate of how important these organisms are in terms of biomass. There are all kinds of different shapes and forms of plankton, as I will introduce you to in a second. But we're interested in these guys because of the roles that they perform in marine ecosystems and in the Earth's biogeochemical cycles. They are the basis of the oceanic food webs. The photosynthetic plankton sequester atmospheric carbon dioxide into the ocean in the form of organic materials or inorganic materials. Through photosynthesis, they also generate oxygen. And because the ocean is deep, a fraction of these organisms, when they die, will sink to the bottom of the ocean, creating what is perhaps the most important biological carbon pump on the planet, the means by which carbon is taken out of the carbon cycle for long periods of time, even geological periods of time. And we know from work performed by many people over the decades that the plankton, on the one hand, can affect climate. They have affected climate during the history of life on Earth. And they're also affected by climate change. So there is a clear feedback between plankton and the climate system. And this is one of the aspects that we would like to learn more about through this program. So what's in your plankton? Let's go through them briefly, just as a setup for the presentation. One very important component of the plankton are the micro-eukaryotes, single-celled eukaryotes, that are also known as protists. All kinds of different shapes and sizes of organisms, as you can see in this micrograph here. Diatoms being in little glass cell walls. Dinoflagellates looking like little alien spaceships, for example, and other kinds of organisms, some of which are photosynthetic, some of which are non-photosynthetic, maybe parasitic, or with other kinds of lifestyles. So there's a whole range of organisms within this micro-eukaryotic community, which I'll touch on a little bit later. And if we add these up, these can represent up to sort of 10 million cells in an average liter of seawater. So those are the micro-eukaryotes, which are very often the primary producers in marine ecosystems. They are grazed upon typically by the zooplankton, which may be like copepods or other kinds of multicellular organisms. And these can be present at up to 100 or so organisms per liter of seawater. And then moving into the smaller organisms, we have the prokaryotic guys, the bacteria, the archaea, some of which are photosynthetic, the cyanobacteria, which can be present at up to 1 billion cells per liter. And then even smaller, we've got the viruses and giant viruses, which infect the other components in the marine ecosystem, which can be present at up to 10 billion particles per liter of seawater. So all of these guys are present in these microscopic ecosystems in the ocean, and we wanted to sample all of these, or we wanted to sample the whole ecosystem from one size, from the smallest to the largest. So that is a challenge in terms of numerical issues, because they're present in different amounts in an average liter of seawater, and they're also very much different sizes. So this is nicely illustrated here with a copepod with a diatom sitting on it and a bacteria sitting on the diatom. Okay, you can see how we're going through different orders of magnitude here, and we don't even have a virus. If there'd be a virus here, it'd be a little particle sitting on top of the bacterial cell. Okay. So we wanted to capture all of these organisms on this sampling expedition. This is about the equivalent of sampling from an ant to a brontosaurus. Okay, so if you think about that, if you wanted to sample a forest, a terrestrial ecosystem, that's sort of the different scales that we have to go through in order to sample this whole marine ecosystem from end to end. So we needed we needed to devise sampling protocols that would allow us to collect all of these different organisms, going from the smallest viruses up through the metazoans of a few millimeters in size, And then we also had to capture the physico-chemical context of the ecosystems that we were sampling. Okay, because obviously without context, it's very difficult to understand much in biology. So these were some of the challenges we faced as we were setting up this project. And then we also realized that we had to do a global sampling campaign, because the ocean is one, it's a single ecosystem. Everything is connected. It may take a few thousand years for an organism to get from here to here, you know, but in principle, everything is connected. So we felt that it was important to sample really the global ecosystem on really the planetary scale. And so this is what we did during, from 2009 to 2013. We left France in September 2009, and we went first through the Mediterranean and the Red Sea, down the Indian Ocean, through South Africa, into the South Atlantic, into the Southern Ocean, then into the South Pacific, out to Tahiti, Hawaii, back to San Diego, through the Panama Canal, up to New York, and back to France. And then in the summer of 2013, we did a second expedition to go around the Arctic Circle up here. Okay, so in that expedition, we went through the Northeast Passage above Russia, and then through the Northwest Passage above Canada, okay, in one season. So Zatara is in fact the first sailboat that has been through both Northeast and Northwest Passages in one season. Sadly, it's possible now because of the ice melt in the Arctic. Okay? But nonetheless, it was it was not without risks, let's say. So so all of this is done now, but just to give you a feel. So from sort of how the science was on board. A lot of the time on board was really spent sampling these different kinds of organisms using different sampling equipment. Could be plankton nets, could be pumps, could be a rosette that we could send down to 1,000 meters to collect water from depth, filtering the seawater, getting the organisms onto membranes of different mesh size, different filter size, to then subsequently be put in the refrigerators in liquid nitrogen and shipped back to the labs. We also had a little dry lab on board with a few microscopes and cytometers with which we could actually observe the organisms that we were sampling to get a feel for what was actually in the water column. And we would also sample at night so that we could catch the movement of the zooplankton as they went up and down the water column. All of this was done at a typical sampling station. We would sit for like 2.5 days sampling all those different kinds of organisms using different sampling equipment and characterizing the physico-chemical environment of the water mass that we were sampling. So in the end, we had sampled at 210 sampling sites. You don't see it too well on the map here, but we'd sampled pretty much using exactly the same protocol at 210 sampling sites around the world, which was great because it means that we can really compare everything with everything, right, which is quite unusual in oceanography, I would say. So from these 210 sampling sites, we had brought back around 40,000 samples for biological analysis, together with other samples, other data sets that would bring in that physico-chemical contextual information that obviously is so important. We typically sampled at the surface, we sampled at the deep chlorophyll maximum, and we sampled in the mesopelagic depths, on average at about 700 meters down in the water column, using these different kinds of equipment to catch all of these different organisms, from the smallest viruses to the zooplankton. And then, okay, you've got the samples, now what do you do with them? The questions that we want to address are really quite straightforward in a simplistic way. We simply want to find out who is there in each sampling site, who is there, what do they do, with whom do they do it, and why. Why is it important for them? Why is it important for the ecosystem? Why is it important for the Earth system and for us ultimately? So to address that simple question, we used the same protocols, very standardized protocols, which were on the one hand based on high throughput Illumina DNA sequencing, also in parallel high throughput imaging of organisms, and thirdly the physicochemical data analysis that we pulled in. So the high throughput sequencing was based on essentially shotgun sequencing of these communities of organisms that we collected and that we had size fractionated into sort of virus-enriched fraction, a prokaryote-enriched fraction, a protist-enriched fraction, and a metazoan-enriched fraction, which obviously had quite a lot of overlap, but at least it helps to decomplexify the genomic information. And then with that, with those samples, we would extract nucleic acids, and we would sequence either just shotgun sequence the DNA to get the metagenomics information. We would isolate messenger RNA and do metatranscriptomics to see the genes being expressed in a particular environment. And then we also did meta-barcoding by focusing on the 18S ribosomal DNA sequence to look at the eukaryotic populations of organisms. So all of this was initiated more than 5 years ago now, and we were very happy that we could publish the first foundational results in a special issue of Science, which was published almost 2 years ago now. This talk's getting old. On May 22nd, 2015, and we were very happy that we could publish 5 papers back to back, describing the foundational results of the project. We got the editorial, we got the front cover. You know, it's a great happens once in a scientist's life, if you're lucky. So this generated a lot of noise, also within the media. We had hundreds of articles written about plankton, suddenly in the daily press. This is the front page of the New York Times. This is their view of what a plankton looks like, but never mind. And so what we reported in those 5 papers, just briefly, in one of them, we described what we call the ocean microbial reference gene catalog, where we basically sampled, we basically sequenced predominantly prokaryote bacterial genes from 68 sampling sites with pretty good global coverage, and generated the ocean microbiome, which consists of about 40 million genes. Okay, that compares to 10 million genes in the human microbiome. So the ocean just has 4 times more genes. This is photic zone. Okay, so this is sort of top 100 meters of the water column. And we also found that the gene catalog is close to saturation. So the more you sequence, the more you just find the same things. So that's more or less the limit of the diversity of genes that we were finding. This gene catalog is about 1,000 times more sequenced than the GOTS project performed by Craig Venter. And of these 40 million genes, about more than 80% were discovered by Tara. And of these genes, as you would imagine, most of them we don't know what they do. They have no annotation or no function. So there's a lot of discovery to be made here to figure out what they do and how they can be used also in certain applications. So that was one paper. In another paper, we performed an assessment of the ocean biome by looking at viral communities, either by quantitative transmission electron microscopy or by doing genomics analyses. And we described in this paper about 5,500 different kinds of viruses. And to put this in perspective, before the project, only 39 were previously known. Okay, so like 99% are new coming from the project. And then for the micro eukaryotes, the protists, things get a bit more complicated because there is so much diversity within these organisms. Most of them we don't have in culture. Many of them we don't even know what they are, let alone having gene sequences for them. So this is why we use the meta-barcoding approach to sort of attempt to put these organisms on the eukaryotic tree of life, which is shown here. And to give you an idea of the diversity here, there are marine representatives in all these branches of the eukaryotic tree of life. The diatoms are here, dinoflagellates are here, for example. But within this branch here, the animals are placed here, and our closest relative are the sponges. So this is the genetic distance between animals and sponges, ourselves and sponges. And you have all this diversity, which we know very little about. So this is why we had to use, we felt, this barcoding approach to begin to uncover the surface of the diversity of these organisms. And what we described for these organisms was about 130,000 genetic types, OTUs, using our molecular sequencing method. Again, this is saturating. So this is more or less, the extent of diversity within this top 100 meters of the global ocean, we would say. And this is more than 10 times. This is more than 10 times higher than the number of formally described marine eukaryotic plankton. So 90% are new, and about a third of them cannot even be assigned to any known taxonomic group. So we cannot fit them in this tree of life yet, either through the limits of the sequence we're using, or because these are really new organisms that we know nothing about. So because there is so much diversity to be uncovered here, we feel it's very important to build up an imaging data set of these organisms. And so to do that, we have set up a high content confocal microscopy platform to be able to generate 3D multicolor images of these organisms in a fairly routine, fairly standardized procedure. And we can generate these kinds of images of the organisms. Here are just 3 examples. This is an Acantharia, where you can see the exoskeleton made of strontium. The chloroplasts are in red, and the DNA is in blue. That's a dinoflagellate. You can see the external shape of the cell with the chloroplast inside. The membranes are shown in green, I think. And here is a diatom, where you can see the silica frustule in light blue. As we dive in, you can see the chloroplasts inside the cell in red, and the nucleus where the DNA is in blue. So we literally have a million of these images like this, and that the challenge is to be able to link these with the one-dimensional DNA sequences in the future. That's a huge project for anybody who likes a challenge. And we're slowly beginning to work on this to be able to use image recognition to identify these organisms, and then to assign DNA sequences to. That's very much a work in progress. But anyway, the sorts of things that came out of analysis of these protists. One study revealed that the Collodaria are incredibly abundant in the ocean. You all heard of Collodaria? Probably. So they're a group of radiolarians. They've been known for a long time. These are Haeckel's drawings of Collodaria. And from the metabarcoding perspective, from the DNA perspective, we didn't find a lot of diversity of these organisms, but we found that they were incredibly abundant. And here we just have we have abundance shown by the size of the bubble, and the occupancy within the different sampling sites that where we sampled, and the evenness of their distributions in all of those sites. And basically, we see a trend towards abundant organisms being very sparse from a diversity point of view. There's only a handful of different genotypes here, but they're all very, very abundant, and all abundant sort of ubiquitously in many of the sampling sites that we looked at. So that's sort of a molecular perspective of these organisms. But we could complement that nicely with an underwater vision profiler that we had on board the Rosette that we would deploy on Tara, which is basically a camera. It's basically a low resolution camera. It has some lights on each side, and it takes images of everything in the water column as the Rosette goes down in the water column. And from those low resolution images, you can then quantify different kinds of organisms, and these are the colodaria that we picked up from imaging directly in the water column, and adding all of these up together, combining this data with other cruisers, we could basically see that that these organisms were as abundant as the zooplankton. At least at mid-low latitudes, the abundance of colodaria was equal to or more than the zooplankton in the water column. So there's a lot of carbon, a lot of biomass in these organisms, which we didn't really know about before we were able to do that survey. So these are sort of the most abundant organisms in the ocean, in the marine planktonic ecosystem. And another surprise is that these guys, the diplonemyids, are the most diverse group of eukaryotes in the ocean. So who's heard of diplonemyids? They're maybe the most diverse organisms on the planet, turns out. So, prior to the Tara Ocean's work, there were 3 known species of diplonemyids. They don't really exist in culture. They are small, sort of amoebas-like organisms, very, very small. At least one of these 3 species, maybe 2 of them, were known to infect diatoms. So that much we knew. But other than that, we really knew nothing about them. They don't have culture. And from Tara Oceans, we could identify 45,000 genetic types of diplonemyids. So we're going from 3 to 45,000, mainly coming from the mesopelagic zone. Okay, so these are deep organisms. What do they do? Well, they're unlikely. Well, they're not phototrophs. We know that they're not photosynthetic. So they presumably have some sort of heterotrophic lifestyle. Maybe they're parasitic, maybe symbiotic, maybe bacteriovorous. We don't know. So there's again a new research territories to be explored to understand what these guys are doing in the ocean, and how come they're so diverse. So that was sort of a summary of different kinds of organisms that we have explored. So we're still very much in the biology space within this diagram here, right? Now I want to move into a little bit the physicochemical context of where these organisms live. So some of the physicochemical data we collected ourselves, and some of it we could bring in from satellite data, SST and salinity and chlorophyll, for example. We could bring in data from the array of Argo buoys that are out there, Argo floats. different kinds of oceanographic networks which are very active in the world's oceans, which are collecting data, which we can use in a contextual way to understand our own data. So we can sort of combine the genomics information that we're collecting with the environmental data from the different sampling sites, pulled in from these different sources, and then we can try and do very large scale analyses to look for correlations, to look for similarities in patterns, and so on. By regression analyses, by co-occurrence networks, and so on. And so one of the very large computes that we did in that line was to put together an ocean interactome. So we have bacteria, we have viruses, we have protists, we have zooplankton from 68 different sampling sites. So we can throw all of that data in together, and we can ask which organisms always co-occur, what is the strength of that co-occurrence, and which organisms never occur together. Okay? So that's a sort of social network of the plankton, and that's the hairball that is generated, which is not particularly nice to look at. But what we did in here is to we looked at taxon associations, and we looked at taxon environment associations to see which were the strongest drivers of associations. And the blue dots here are eukaryotes, and the pink are the bacteria, and so on. And the green edges are co-occurrences, and the red edges are the opposite, sort of mutual exclusions. Okay, so we can see some hubs, some plankton hubs, organisms that are very central, which may be in a sort of negative way, like some guys over here that are pretty central. This one here, but are typically excluding other organisms. Okay, you can think of these as the Putins, the Trumps of the plankton world, right? You have other, sort of universally positive plankton, which would be, well, in Boston, I'd say Tom Brady. But I don't know who I'd say in UNC. Right. So what emerges from this, if we compare the taxon and the taxon environment interactions, it turns out that the biotic interactions are far more abundant than the abiotic interactions. We're a bit surprised to find this. So this basically tells us that what is important is it's who you live with. That's more important than where you live. Okay, so it's the community that you live in that is more important than the temperature you live at or other parameters. Okay, so the biology is driving the interactions, which we were quite surprised to see that signal. Then what we also observed here is if we pulled apart those positive associations and the negative associations, we could see that the positive associations far outweigh the negative associations, telling us perhaps that cooperation here is more important than competition. So sort of who you live with, working together to ensure that the ecosystem as a whole works together, being perhaps more important than just survival of the fittest, competing, trying to screw your neighbor, sort of thing. Again, we were quite surprised to see that signal in coming from the plankton world, which is not necessarily the way that we view evolution. We tend to think of evolution as being about competition, about survival of the fittest individuals and so on. But maybe it's more also, the community that you live in, ensuring that community has is robust and is able to move forward together. That is important. So we were surprised to see that. We were also surprised to see that this signal is actually not always true. And a big exception to this are the diatoms, which turn out to be the antisocial guys. So if we look at these co-occurrences and we look at the exclusions, just for the diatoms and who they interact with or not with other groups of organisms, we find that the co-occurrences are pretty few and far between, whereas the exclusions are significantly greater. They're telling us that sort of in this anthropomorphic way, diatoms are few friends and they're able to keep their competitors at bay. I think sort of chimes with our idea of how diatoms hang out in the oceans as being typically going through these boom and bust proliferation cycles, being often the first on the scene when nutrients, right, when everything is ready, the diatoms will often be the first to take over, to take off. They'll proliferate, they'll dominate, and they will only leave space to the other organisms, to the other phytoplankton, when the silicate is run out, for example. So it sort of chimes, I think, with some of the things that we think about diatoms. We can also see this if we convert this into a sort of food web perspective of the interactions. And we look at different kinds of phytoplankton, and we look at different kinds of zooplankton, organized into plankton functional types, the PFTs. And here again, we're expressing positive associations by green edges and negatives by red. We again see that the silicifying phytoplankton, which are by and large the diatoms, have these negative signals, whereas the other kinds of phytoplankton do not. Okay, so using these different methods, we can get this this exclusion signal, which, as I say, sort of chimes with our idea about diatoms being often the first to bloom and so on. So that's the sort of thing that we can glean out of this interactome. It is also a playground to discover new interactions. So this is a computer-generated figure, right? We have no clue whether these interactions really occur in the ocean in real life, but we can use this as a sort of playground to explore interactions. And here's one example of that. From Symbiodinium, this super dinoflagellate, symbiotic with all kinds of different organisms, although organisms that are typically benthic, like corals, like different kinds of metazoans. The Symbiodinium is really a champion of symbiosis in many ways that we know about, but we don't know anything about symbiosis of Symbiodinium in the plankton, in the pelagic world. Okay, so this is what we started to explore in the interactome, and we found out that Symbiodinium in the plankton tends not to live by itself, but it lives inside another organism. It lives inside this little ciliate, Tiarina, little hairy ciliate here. And here's the Symbiodinium inside this ciliate. And this is probably, how it transported around the ocean and maybe colonizes coral reefs in different areas and so on. So we picked this up from the interactome. We went and saw the ubiquity of this interaction in different areas around the ocean. And then through the imaging data that we have, we could actually physically see the interaction of Symbiodinium living inside this ciliate single-celled organism. So that's one example of how we can explore this interactome and find new interactions. I think in the interest of time, I'll miss this one. And move on to another topic, the ocean's biological carbon pump, which I briefly introduced you to earlier, which is, as many of you will know, concerns the generation of organic material in surface waters in the photic zone of the ocean, followed by its consumption or eventual sinking down to the sequestration layers of the ocean. where it can be removed from the carbon cycle for very, very long periods of time. So our sort of understanding of this biological carbon pump, if we think about the fluxes of organic material from the photic zone down to the deeper layers, we sort of the data indicates that the larger phytoplankton, like diatoms, for example, tend to promote downward fluxes of carbon to the ocean interior, as does degradation from zooplankton, being perhaps more important than microbial degradation. This is sort of our view of how the ocean's carbon pump may work. Now we can try and address this in the Tara data set, because if we use this underwater vision profiler again, so as this is lowered down into the water column, we take images of all of the particles in the water column, which you see here. So this is sort of going down the water column. You can see all these particles that we can take images of using this system, and then we can calculate the amount of carbon, the amount of aggregated material in the water column, and can convert this information into carbon export using Stokes' law. And this is sort of the view of carbon export that we get when we do this, going through the Tara Oceans transect. Okay, so, and here we're looking at export. We're looking at particulate material down to 500 meters in the water column. And you can see some areas have a lot of material in the water column down in the Southern Ocean, for example. Other areas are pretty clean, pretty transparent. South Atlantic, South Pacific, and so on. So we can take this sort of data and we can combine it with this sort of data and figure out, so who are the guys that are correlating most strongly with the carbon export that we see using the UVP. So we do that using different computational tricks that I'll not really go into. But basically, what emerged from this work is that the organisms that correlate most strongly with carbon export depth from this Tara Oceans data set are radiolarians, dinoflagellates, Synechococcus, and the viruses that infect Synechococcus, the Synechophages. So these came out of independent studies, and we were surprised to see that the small guys seem to have quite an important role here, not necessarily the big guys, and perhaps viral infection, in particular Synechophage infection of Synechococcus, is maybe instrumental in creating this flux of carbon to the interior. So these analyses are incredibly predictive. We can in fact predict up to 89% of carbon export with just a handful of organisms. So if you give me a sample of seawater, I look at those organisms, I could predict the export with 89% of chance of being right, sort of thing. So it's quite remarkable that it works to that extent. And I think it's also a nice example of how you can use these approaches to sort of try to scale all the way from genes all the way up to ecosystems by bringing in these different data sets and looking at correlations between them. Finally, I just want to introduce you to some new work, which is still very much a work in progress, where we're looking at the eukaryotic genes. Okay, so we have the prokaryotic gene catalog, the ocean microbiome, 40 million genes, saturating, quite a nice data set to work with. We also obviously wanted to get a eukaryotic reference gene catalog too, in spite of all the challenges associated with that. So we basically used the same 68 sampling sites as we did for the ocean microbiome, and whereas we get saturation of the prokaryotic gene catalog at about 40 million genes, the eukaryotic genes just keep going up and up. So we're at 115 million different genes, and we're still climbing. Okay, so that's enough to stop. So that's the extent of the diversity that we have to deal with within these eukaryotic genes. You don't see this too well. 50% of them, we have no clue what they are. They have no matches in any public databases anywhere. About 30% of them we can trace back to the MMETSP sequences from the Moore Foundation, which shows the value of that effort from the Moore Foundation. You see the organismal distribution there, which I won't go into too much. And so basically, comparing our data set with the Moore Foundation's transcriptomes, about 30% of our sequences do have a match in MMETSP, and about 60% of the MMETSP sequences have a match in Tara Oceans, which is not too bad. It certainly shows the value of the Moore Foundation's work. If we look at which organisms are matching the best between the two data sets, it's often the Mammaliales, Bathycoccus, and so on, but you also see some dinoflagellates, some diatoms as well, that are top scorers in the eukaryotic data set. And at a very, very large global level, we can sort of look at these 115 million genes and ask, well, what are the most highly expressed genes? This maybe is the first question you would ask. And here we're using protein domains based on PFAM. We have the top 100 protein domains divided by size fraction, divided by taxonomy, and divided by oceanic region. And without going into any details, let me just say that motility comes out as a pretty strong signal, even in single-celled organisms. So these guys seem to spend quite a bit of energy moving around. Contact with their environment, extracellular perception, intracellular signaling. I was surprised to see such a strong signal from this very large overview of the data. Translation, okay, and parasitism is also a strong signal. There are some protein domains which we believe to be specific for parasites coming from trypanosomes, leishmanias, plasmodium, things like that, which are turning up quite commonly in the ocean, perhaps telling us about the importance of parasitism as a strategy, as a life history strategy in the ocean. So, and then finally, let's see if we can do anything with this data. All right, so it's 115 million genes, it's overwhelming, but we are slowly figuring out how to work with the data. And here are just some simple analyses where we just try to ask some simple questions. And here we're looking at silica transporter genes, which are specifically coming from diatoms. So we have no real taxonomic issues for where these genes are coming from. We know that they're only from diatoms. And we can try and link the abundance of silica transporter genes in these different sampling sites with silicate concentrations that we measured at those different sampling sites. And we basically see that, yeah, it works. So the higher the measured silicate concentration, the lower the expression of these SIP genes in the environment. So it does follow what we would expect to occur. And a second example are the ISIP genes, which many of you will know are iron responsive genes, which we can again trace back to diatoms quite easily. Here we're looking at abundance of ISIP transcripts, again in these different sampling sites. And we're correlating the mRNA levels to modeled iron concentrations, 5-year averages. And again, we can see that as iron concentrations increase, ISIP expression goes down, which again is what we expect from from the experiments of Adrian's lab and others. But I think it's fantastic. Here we are looking at gene expression in communities in the ocean. These are not bottle experiments. They're not, cultures in the lab. These are real communities living in the ocean in a natural context. And I think the fact that we can see these signals is very encouraging for future work. So that's just sort of the start of this work, and I'll be happy to talk more about it with you later, either together or individually. So I will end here in the interest of time, and to summarize what I've talked about then. I think the value of Tara Oceans is that it's the first end-to-end description of a global ecosystem, going from the viruses all the way up to the zooplankton in a global ecosystem, which is the ocean. There's no real example of that sort of thing prior to our work. This is also possible because the ocean is a simplified system. If you wanted to do this in soil, if you sample soil here and sample soil there, it'd be two different worlds. So the ocean is simple compared with soil, for example, terrestrial ecosystems. So I think that has helped us a lot. The ocean microbiome consists of about 35,000 different kinds of bacteria that are mostly known, containing about 40 million genes that are mostly unknown. Viral communities that we described are on the order of about 5,000. About 99% of those are new. The diversity of eukaryotic plankton is huge at about 130,000 OTUs, but it is finite. We can see it's coming to reaching saturation. And about 90% of these are new, and there's a considerable unknown component. I also spent a bit of time with the plankton interactome, showing you a predominance of co-occurrences over exclusions, with diatoms being an exception, and gave you an example of how we can use this as a discovery ground to find new kinds of interactions. And then finally, the eukaryotic reference gene catalog is slowly being made tractable at 116 million unigenes with no sign of saturation, and that we're beginning to do differential gene expression analysis. And finally, all of this data is public. You are very welcome to use it. And you better start using it soon, because what we've done so far is only from 579 samples. So better to start using it now rather than waiting until all 40,000. So with that, I will end and encourage you to explore this data set. And I'll thank the people involved in the work, with obviously a large consortium of different labs. We're about 23 different labs, including Mick Follows at MIT, Matt Sullivan, formerly at the University of Arizona, the University of Maine, Mike Siraki, and the Single Cell Genomics, proud of that. Our sponsors, our funders, who've been essential, so thanks to them and thanks to you.